It's Professor Dave. Let's talk about viruses.
He knows a lot about the science stuff. Professor Dave explains.
We just spent a number of tutorials learning about pathogenic bacteria. Back in the late 19th century, scientists could physically observe many of these infectious agents under a microscope and even grow some of them in the laboratory. But in the 1890s, two scientists named D.M. Ivanovsky and Martinus Beijerinck were puzzled by an unusual infectious agent. They observed a disease in tobacco plants called mosaic disease, but the infectious agent was too small to be seen with a light microscope. They could only grow this agent in media that contained living cells, and to their surprise, it could pass right through the filters they typically used to catch bacteria. Beijerinck called the mysterious infectious agent a filterable virus, where the term virus comes from a Latin word literally meaning poison. About 10 years later, two other scientists named Félix Durel in France and F.W. Twort in England independently discovered such a filterable virus that infects bacteria. A virus that infects bacteria came to be known as a bacteriophage, and with that, the field of modern virology was born. We learned a bit about viruses in the biology series, so for some background information, visit this tutorial first, as we will soon be investigating a variety of specific viruses in quite a bit more depth. To summarize a few key points from that lesson, a virus is just a bundle of genetic information, which is either RNA or DNA, but never both, contained within a protective protein coat. Viruses are inert particles, meaning they can't move around on their own. They're also incapable of metabolism or replication on their own. When a virus finds its way to a host cell, however, the virus hijacks the cell's machinery, using the cell to make more copies of itself. Viruses are considered obligate intracellular parasites, and most scientists agree that viruses are not alive, as they don't fit the criteria we use to define life. Viruses can be grouped into two general categories based on the type of cells they infect, those being prokaryotic and eukaryotic. Although both groups are viruses, as we said, the ones that infect bacteria are referred to as bacteriophages, or sometimes just phages. Now, how big are these? Most viruses are 100 to 1,000 times smaller than the cells they infect, which explains why early microbiologists could not see them. They can range from about 10 nanometers in diameter, with as few as 10 genes, to about 800 nanometers in diameter, which is quite a range. Let's introduce a few more important terms. As we said, at the bare minimum, a viral particle, also called a virion, is comprised of a nucleic acid surrounded by a protein coat. The protein coat is called a capsid, and it protects the nucleic acid from any chemicals or enzymes the virus might encounter. Together, the capsid and the nucleic acid it protects is called the nucleocapsid. Some viruses have a lipid bilayer outside of the capsid, which is called an envelope. Viruses without an envelope are called naked viruses. Phages, in fact, are nearly all naked. Since all viruses contain a single type of nucleic acid, they're first classified as either an RNA virus or DNA virus. Some viruses have a circular genome, while others have a linear genome. The genome itself can be single-stranded or double-stranded, and the type of genome that a virus has informs its replication strategy. Generally, viruses fall into three shapes: helical, icosahedral, or complex. Helical viruses look cylindrical when viewed with an electron microscope and can be either short and rigid or long and filamentous. Icosahedral viruses look spherical under an electron microscope, but their surface is actually much like that of a soccer ball, with 20 flat triangles arranged around the surface. Complex viruses are more intricately constructed and tend to vary more in shape. Viruses also have specific protein components that enable the virion to bind to receptors on the surface of host cells. More on that later. When it comes to viral classification and nomenclature, it can get pretty messy. The good news is that there's an entire international committee dedicated to classifying viruses, called the International Committee on Taxonomy of Viruses, or ICTV. According to this system, a species of virus is defined as a population of viruses that share a pool of genes distinct from the gene pools of other viruses. So even though we don't consider them to be alive, viruses can still be grouped into a species. Species of viruses can be grouped into a genus. A group of related genera, which is plural for genus, can be grouped into a subfamily. A group of related subfamilies can be grouped into a family. And finally, a group of related families is called an order. For the most part, classification into families is based on characteristics of the virion and its genome. The criteria are as follows. First, there is morphology. What is the size and shape of the virion? Does the virion have an envelope? This is all usually determined by electron microscopy. Next, we have all of the structural proteins. What is the molecular weight of the virus? And lastly, there are genome properties. What type of nucleic acid does it have? Is it double-stranded or single-stranded? If single-stranded, is it positive sense or negative sense? What is the sequence of nucleotides? These are the relevant criteria that are considered. Unfortunately, the names of viruses have no consistent pattern other than the names of virus families ending in the italicized suffix viridae. Some are named for the appearance of the virus, some are named for the geographic region they were discovered in, some are named for the disease they cause, and some by their route of transmission. While bacteria are typically called by both their genus and species name, viruses are typically referred to by their species name only. And to make matters worse, many virologists refer to viruses by informal names. We will be looking at a number of these viruses moving forward, so now that we've recalled some of the basics, let's move forward and talk about what happens when a virus encounters a host cell. Thanks for watching, guys. Subscribe to my channel for more tutorials. Support me on Patreon so I can keep making content. And as always, feel free to email me, ProfessorDaveExplains@gmail.com.